its kind of thick structure in this EM picture over here. If you were to cut it kind of right in the middle and then look down the barrel, that's what it would look like. In how it is shaped. And so you can see that it has a lot of these tubulin structures, just kind of, actually no, this is a different structure, primary cilia, which is microtubule based. And so each of these is a microtubule, as you can see here. And then there are just concentric rings around that. But you can see microtubules, again, extending across a lot of the cell, similar to actin. And so we'll get into why it's so different from actin. So today we'll be talking about viruses, how they travel within cells. We'll talk about kinesins, dynamic instability, and also talk about microtubule-associated proteins. Okay? So this is herpes simplex. It's caused by a virus. This infects lots of people. Probably 80% of the people out there have the herpes simplex virus. You don't always get cold sores, but you could be infected and not show any sort of phenotype. This is the infection mechanism cycle. Here are the viruses floating around. You get exposed. Typically your skin cells, the epithelial cells, get exposed. And those viruses will then go ahead and infect the epithelial cells. And then the genetic material incorporates into the DNA. And so now that host cell has viral DNA incorporated in the genome, such that when it replicates, it will still have that viral genome. It could produce more viral particles that are then released from the cell. And in this case, it can actually infect neurons. Your skin cells are actually innervated by neurons. That's essentially how we're able to sense light, sense touch, and amongst other things. And so then the viral capsid will travel down into the body of that neuron, and it will, again, incorporate into the DNA. And then that's the latency. So you can get infected through this cycle, and it lays dormant here for years. You may not even know that you're infected when you're in the latency phase. But then the virus has to reactivate. Oftentimes, some sort of stress triggers it. It could be you stressing out over the exam. That's happening on Wednesday. It could be you ate something spicy. Or it could be that you're slightly allergic to something. Whatever it is, you could reactivate the transcription and translation. It could produce viral particles, and it could reinfect the skin. And in this recurrent infection is your cold sore. That's popping up. And then when it eventually goes away, after weeks and weeks, it's now back in the latency phase. Okay? All right. So that's essentially a small version of how viruses infect us and how they just kind of stay dormant. Now here comes the first question. Blank helps the capsid travel along microtubule tracks towards the nucleus, whereas blank helps the capsid travel back towards the plasma membrane. All right, so over 80% of you correctly talk about kinesins and dyneins, but about 50% of you say C, which is dyneins help the capsid travel towards the nucleus, whereas kinesins towards the plasma membrane, and then About 30% of you say the opposite. So what is the correct answer? And it's C, dynein and kinesins. So let's talk a little bit about them. So here is that neuron. Here's that epithelial cell, all the viral capsid particles being produced, and then infection. And so in order for that capsid to then travel along towards the nucleus of the cell, it has to undergo retrograde transport. Retrograde transport is transport towards the minus end of microtubules. And it uses dynein to perform that minus end transport, right? And so you can see that this capsid particle could potentially interact with both kinesins and dyneins. But when it wants to go retrograde, it's only really using the dyneins to go towards the nucleus. That's where it gets over there. It could dump the genetic material into that nucleus and then incorporates into the nuclear genome. And then vice versa, it'll use kinesins once you are producing viral capsids and you want to then get them released. It'll use anterograde transport towards the plus end. And then kinesins are used in this process. Okay? And so there are two motor systems, and they do walk along microtubules in opposite directions. And so what this lets you know, in a way, is that microtubules are the highway of the cell. In order to move things around in the cell, be it vesicles, some proteins, organelles, they will use microtubules to facilitate their transport. If it was a large vesicle, like here, this is a viral particle, but if it was a larger vesicle, it would have these things attached so that it could march along and then move along these highways. Okay? And so what you'll see is that every cell essentially has a microtubule organizing center. That microtubule organizing center is made up of the mother and daughter centriole. And then this is where dozens, hundreds of microtubules will nucleate from. And so their nucleation will allow them to create these long stretches of highway towards different parts of the cell. And in this case, like a mitochondria can move back and forth along these microtubule highways, endosomes, Golgi, ER, in addition to viral particles. Right? Because it's much more efficient to use a highway than it is to just randomly float in the cell hoping to get to the direction that you want to be in, right? And all of this is facilitated by these motor proteins on microtubules. And so this is what it looks like. This is a movie where different vesicles were actually marked with a color, and you can see them being transported along at different speeds along these microtubule tracks. So you can see some of the smaller ones are incredibly fast, some of the bigger ones are a little bit slower, but they're all being marched along in very specific directions, right? And it really does look like a highway system. Okay? And the last part is just a fancy way of just showing the speed at which some of these particles are moving. All right. So let's get into how some of these motor proteins actually work. And so the first one we'll talk about is the kinesin cycle. We'll be talking about dynein next lecture. But essentially they walk in a hand-over-hand motion. And so they essentially don't let go of the microtubule. Meaning there's always one hand attached to the microtubule. Because if you didn't have it in that way, you could potentially fall off the microtubule, right? And so you always have to have one hand or one motor protein always touching that microtubule or you'll fall off. And so this is the process denoted here. So the rear or lagging head is tightly bound to the microtubule and to ATP. Okay? When it's ATP bound, it's tightly bound to the microtubule. ATP hydrolysis of lagging head leads to weaker binding. So you want hydrolysis so that you can start your step. Because if you were bound to ATP, it's like having glue at the bottom of your shoe. You're not going to really be lifting it up. Okay? The front or leading head is loosely bound to microtubules and ADP. So this is the one that's after hydrolysis. And the ATP binding leads to to the leading head, induces this linker region in purple to shift from the back end of that motor to the front end, essentially pulling it forward. And that's the walk. Okay? And so ATP is bound on the lagging head. ADP is bound on the leading head. Once this is hydrolyzed, you're poised to step. ATP addition to the leading head causes a conformational change that essentially flips the lagging head in front. And then that is the first step. Then this is hydrolyzed. ADP is exchanged for ATP. You make another step. Okay? Now, this is kind of what it looks like. This is just a cartoon of kinesin essentially walking. So you can see kind of its weird looking gait. ATP bound, ATP bound, ATP bound, and then hydrolysis on the back end, hydrolysis on the back end in order to make that step. If they're both bound to ATP, it wouldn't make a step, right? If they're both bound to ADP, wouldn't make a step. You need hydrolysis on one end, and then you need ATP binding on the front end. And you can see that this sucker can really attach to very large things as it's moving. So if it was a small particle, like in that movie where we're looking at the trafficking, it can go pretty quickly. If it's a large sucker that's, again, being prevented by a lot of the viscous solution, that force behind it, you're going to move a lot slower. Okay? And so that is the kinesin cycle. Do you have any questions when it comes to this? Yes, in order for this step to occur, there has to be constant exchange of ATP molecules in order for you to utilize that energy throughout this whole process.
So on that second picture where the hydrolysis just occurred on ATP,
Yes.
so in that moment, the interaction with that back leg or that foot, it's still kind of interacting with it, but it hasn't fully let go of it, which allows this like time
Yeah.
where it's kind of full.
Yes, it's a much weaker interaction. It can pop off much quicker. And so when the front head binds to ATP and you get that conformational change, you're essentially ripping it off because it was a super weak interaction. You're unbound. And then when you flip over to the front, you have a weak interaction on the front. All right. So, how does ATP binding affect the kinesin motor cycle? You can see is that about 65% of you say D, which is both A and C are used for ATP binding. Allows the kinesin head to tightly bind to microtubules, induces a conformational change in the neck linker to shift the lagging head forward. And that is correct, right? ATP binding essentially allows for both of these things to occur. Okay? Good job. Now, back to viruses. Here's the herpes simplex virus, and it's actually entering into the cell. So these are different images, real images of the herpes simplex virus. This is the plasma membrane. Remember, the plasma membrane is really wavy, and it's not necessarily all straight and concentric. But you can see that it ends up docking, and then it gets pushed in, the capsid gets pushed into the cell, and then it starts marching along towards the nuclear envelope, and you can see that it's essentially dumping its material into the nucleus once you're there. Okay? And so this is the actual viral infection, and it's utilizing microtubules to end up walking towards the nuclear envelope. When you look at this, essentially this is, here's a microtubule organizing center. These are different viral capsids in bright white. And so when the cell's infected, here's some ones that are just entering into the cell. They're going along all these microtubules. And here you can see that they're locating around the nucleus because it wants to inject your DNA in there so that you can have viral DNA here. And here's a microtubule in close-up picture with the capsid around that. And then this little thing right there would be the dynein, bringing it into the nucleus. Okay. So what most likely happens when Taxol, which is a microtubule stabilizing drug, is added to cells infected with the herpes simplex virus? Okay, so what you'll see is about 40% of you, 35% of you say E, which is virus infections normal. 20 to 25% of you say C and B, which is enters the cell but stays at the plasma membrane, enters the cell but wanders aimlessly. Okay, so what do you think the correct answer is? Well, why don't you retake this, talk to your neighbor before you click in again, and then click in. All right, go ahead and click in if you haven't already done so already. So still about 45, almost 50% say E, 25% say B, and those seem to be the more dominant answers. And so what is the correct answer? E, virus infection is normal. So if you have a microtubule stabilizing drug, that's essentially like putting a shiny new coat onto a highway, right? All you're doing is making it a better highway. You're not allowing it to disassemble. And so being able to traffic along that highway is going to be unchanged. It's just going to still be there, and it'll still allow the virus infection to be normal, to allow the trafficking of the capsid towards the nucleus. Because all you're doing is stabilizing the highway and preventing it from disassembling.
Could you also say that it's really like a situation where the microtubule is going to be longer than normal because it doesn't have the ability to disassociate on that?
It might be a little bit longer than normal, but the highway will be there.
Yeah.
Mm-hmm. So we're not saying that the microtubules won't be affected, but what we're saying is that trafficking along the microtubules in this case won't be affected. Okay? Yes?
If it was destabilizing the microtubules, would that then be C?
Well, if it's destabilized the microtubules, you don't have a highway system.
Like completely, you're going to assume that with.
Yeah, no highway system, right? No way for the capsid to move around.
So would it just stay in the.
Well, I would probably have to change some of these answers so that there isn't confusion as to which one you should pick.
Okay. So what would be the answer?
Well, there's a solution in the cell. It's a big like ocean. And so if something falls into the ocean, are you going to stay at the surface of the ocean, or are you going to potentially move around with the waves and stuff in the ocean? And that would be your answer. Okay, and so here it is, viral infection in the presence of Taxol. So this is the nucleus. You have a bunch of viral particles surrounding the nucleus of all these cells. And then here, when you add Taxol, and then you ask whether these little viruses can actually get to the nucleus, they can. They're still surrounding the nucleus in this case, okay? And so Taxol won't abolish the ability of these capsids to traffic to the nucleus. Now, here's the next question. What most likely happens when cytoclasin D, a barbed end actin blocker, is added to cells infected with the herpes simplex virus? All right, what you see is about 30% of you said E, which is viral infection is normal, and about 25% of you say both B or C, enters the cell but stays in the plasma membrane, or enters the cell but wanders aimlessly. And so what is the correct answer? E. E, viral infection is normal. Essentially, actin will not be influencing that highway. If you block actin polymerization, if you promote its disassembly, if you promote its assembly, you're not going to influence the ability of the traffic to go along these microtubule shafts. Microtubules don't need actin to polymerize, and these motor proteins don't need actin to operate. Okay, and so if you influence actin, you're not going to influence viral infection. And this is what it looks like. So here's cytoclasin D added to the cells, and you're still getting capsid, viral capsids localizing around the nucleus in this last picture. Okay, and so you're still able to get there. Okay, so now let's talk a little bit about microtubules themselves. And microtubule is a heterodimer. It's actually two tubulins connected to each other, and they're bound to GTP as its energy. And so the alpha tubulin binds to GTP, but it's never hydrolyzed. The beta tubulin is bound to either GTP or GDP. And whether it's bound to GTP or GDP influences its shape. So here's Taxol. This is just that drug that can bind and stabilize these microtubules. Okay. And what you can see here is this is the protofilament being formed. And so you can see that there is the filament system essentially is polarized, where the alpha GTP-bound subunit is on the back end, and then on top is the beta tubulin. Okay? And so there are 13 polar protofilaments to generate this microtubule. So if you were to count this circle, there'd be 13 of these protofilaments, which are along here, 13 of them. And that forms essentially that microtubule cylinder. Okay? Now, I would love for you to discuss amongst yourselves, how is the energy required for microtubule assembly similar to actin polymerization? What are some of the commonalities between these two filament systems? Okay? So turn to your neighbor and please discuss that. Is there a Brian Kaji in the room? No Brian Kaji? All right, just curious. Anyone want to use Brian Kaji's credit card to buy something? So anyone want to venture a guess as to how the energy required for the assembly of microtubules is similar to the assembly of actin?
So actin uses ATP hydrolysis to polymerize, and then microtubular uses GTP hydrolysis. So both using a type of hydrolysis to grow.
They both use a type of hydrolysis, but it's not to grow. So the hand over hand is going to be specific for that kinesin motor system.
You need the energy source in order to find which points the stable interactions, and as the hydrolysis of the energy source happens, it now is going to convert into like a GDP, which is less stable, which allows the dissociation in the back end. So now that process is going to allow it to grow.
Yes. So the binding of ATP or GTP allows you to have a subunit that's capable of assembling. It is added to the filament so that it will grow. And the hydrolysis of those subunits gives it a bad shape so that it's a weaker interactor and you will disassemble, right? And so even though they use different energy, this one's GTP and Actin's ATP, the principle is the same. A good conformation to grow, bad conformation to disassemble. Okay? And that's shown here. So here is a GTP-bound tubulin dimer. So we're no longer looking at the GTP-bound alpha. We're just looking at the beta, since that's the one that hydrolyzes. And you can see that in this GTP-bound form, you're essentially in a good conformation to interact with others. And so you could quickly go ahead and add onto this protofilament. But when you're GDP bound, you kink a little off to the side, and you can fall off. And then you need that GDP-GTP exchange in order to recycle and then go and grow as part of the filament again. And so here is a growing filament. You have the 13 protofilaments, and you have the tubulin dimers being added in a spiral. But then the new addition, they're all bound to GTP, right? But then over time, it hydrolyzes, and then you get into the bad conformation. And once you've lost that GTP cap, say you no longer add anymore, the hydrolysis reaches the tip, you end up shrinking. You end up undergoing catastrophe. These just fall off. Okay? So you can think of this as Legos in a way. When it's GTP bound, you have a beautiful Lego that you can create a beautiful structure with. Right? But if you keep that Lego out in the sun for too long, you bake it on your frying pan or something, and it essentially melts a bit, it's not going to be used very well to build that structure. And so you can imagine that, you know, someone is lighting a torch along the side here, that's the GTP hydrolysis, and then eventually that whole sucker will just kind of break apart. Okay? Now, any questions when it comes to microtubule assembly or disassembly? Why would you need to what? Okay, so why would you need to disassemble microtubules? Anyone want to answer that question? Certainly, you disassemble microtubules and you use them during mitosis, absolutely. But in general, conceptually, why would you want to disassemble a microtubule? You don't want certain processes happening all the time. You don't want the highway system to be static. If you're a cell that is constantly moving, you have to keep remodeling your highway system to end up trafficking things to the proper location, right? And so you need to be dynamic. If this was static, the cell would be stuck. It couldn't really deform its membrane and move as it should. Right? And so you need to very dynamically grow the system and destabilize it. And we'll use mitosis as a fantastic system to illustrate this in the upcoming lectures.
I was reading that this process is also like concentration dependent. Mm-hmm. So can it also go through like a treadmilling phase similar to like actin?
So we'll talk about that. Yes. So microtubules rarely treadmill. They can treadmill, and we'll talk a little bit about that treadmilling process during mitosis. In most instances, it won't, and we'll get into why.
So in the slide before that, you said that the alpha tubulin always had GTP, right?
Uh-huh.
So here we're just looking at the beta tubulin.
Yep.
We just keep in mind that the alpha tubulin has GTP. Does that have a use? Or is it just there?
It's there for structural purposes. It's a tubulin subunit, and so in order for it to have a proper structure, it needs GTP to be bound. All right, so just like actin, microtubules have a lot of accessory proteins that do a lot of things. They have ones that, for instance, sequester those subunits so that they can't be added onto the growing microtubule. You have ones that are nucleators to help promote the polymerization. You have things that will attach the microtubules to other structures such as the membrane. You have proteins that stabilize or destabilize. You have proteins that sever microtubules. You have ones that will bundle these microtubules. And so just about any protein you could think of for actin, there's likely a microtubule counterpart. They're very similar structures in how they are assembled and disassembled. Okay? And so you need a lot of these proteins to help with that process. Now, this is the microtubule nucleator. And it's called the gamma Turk. It's made up of 7 gamma tusks. And each of these tusks have 2 tubulin monomers that associate with accessory proteins. And so this is what it looks like. So here's the 2 tubulin monomers. And then you have essentially 7 of these in a concentric circle. So here's the gamma tusk. And you can see that that 14th one sits on that first one. So that you actually have a spiral. Because this is a spiral system that ends up going up. There's 13 of these, with number 14 sitting on number 1. And then it allows these tubulin heterodimers to then assemble on this growing filament. Okay. Do I have a question about that afterwards? Nope. Why do you think most microtubules need a nucleator? We talked a lot about actin. Thrown in a test tube, you undergo this lag phase, and then once you get past that lag phase, you start growing. If I were to throw a lot of these heterodimers into a tube, that lag phase would be a lot longer. And you may never actually reach that part where you're actually growing.
So to be more specific about what it means with the microtubules.
Okay, so maybe you have a bit more complex of a structure, and so the cell wants to be able to control it more so than a microtube, than an actin filament. Quickness and efficiency, okay. And so maybe you want to be able to control them differently and have one be a more efficient process than the other. Okay, super large energy barrier. How many subunits for actin do you need to make a stable nucleator, a stable seed? 3. How many do you think you need for a microtubule? How many subunits do you think you need to make a stable nucleator for a microtubule? Probably around 14, which is what this sucker has, right? Because keep in mind that, see how these protofilaments are stacking on top of each other? Alpha, beta, alpha, beta, alpha, beta. If you're just assembling that in a straight line, the only contacts you have with the other heterodimer is right there between the beta and alpha subunit. But if you have this system, this nucleator set up, you can have attachments to the side, the bottom, and then when the next one attaches, you'll have it on the other side. So you have far more interactions to keep this stable when you have that nucleator present. And in order to get 14 of these suckers individually attached to each other to form this base nucleator is a huge energy barrier. It's tough to have all those things just happen to interact with each other in just the right way. Okay? And that's why most microtubules need a nucleator. All right. And so here is the centrosome consisting of the mother and daughter centriole. And so this is the microtubule organizing center. Here's a big cell. Here's the nucleus. That little dot is the centrosome. All of this red surrounding it are all the microtubules that are being nucleated from that dot. It's crazy. Essentially, here it is, the mother and daughter centriole. And what you'll see is that there are gamma Turks all, this is the gamma Turk, right? Gamma Turk, all inside this cloud, nucleating all of these microtubules that are radiating. So plus-ended going out, minus end surrounding this cloud. And so you have all these proteins, all of these nucleators sitting right there in that cloud, and that's why it's called the microtubule organizing center, because all the microtubules will then nucleate from that sucker. Okay. All right. So here are some examples of microtubule binding proteins. Here's the microtubule. It has a GTP cap. If you want to destabilize a microtubule cap, you could have some proteins, like for instance, this is a kinesin 13. It can bind to the GTP-bound form of the heterodimer, and it peels it away. And so if you peel enough of it away until you get to the GDP-bound portions, it'll then enter catastrophe. Alternatively, you could have a stabilizer that are specifically binding to that GTP-bound cap, or maybe even binding along the microtubule shaft, and these will stabilize the microtubule. Okay? And so again, a very similar process when it comes to actin. And that's it. Thank you very much.